Type 2 Diabetes by Lily Adams, Evana Darby, Canah Dawson, and Eva Jones. Diabetes is a disorder characterized by abnormally high blood sugar levels. In type 2 diabetes, the body stops using and making insulin, a hormone produced in the pancreas that helps regulate blood sugar levels properly. At high levels of blood sugar, the body releases insulin and adjusts how much glucose enters the cells. However, this process is halted by lifestyle or genetic issues. Type 2 diabetes impacts many parts of the body, including major organs such as your heart, blood vessels, nerves, eyes, and kidneys. According to 23andMe, genetics make up 26% of type 2 diabetes risk. As a result, the environment is incredibly impactful to one's risk of developing diabetes. Some environmental factors include the healthiness of one's diet and whether they live an active or sedentary lifestyle. There are approximately 150 known genetic variations that are linked to type 2 diabetes. Our test would account for all of the mutations that are SNPs. Two of the main indicators of increased risk of developing type 2 diabetes are RS7903146 and RS1326664. The former is from TCF7L2 gene and impairs the insulin secretion and incretin effects and enhances the rate of hepatic glucose production. TCF7L2 expression in human islets was increased fivefold in type 2 diabetes, particularly in carriers of the TT genotype. The second SNP is from the SLC30A8 gene expressed in the pancreatic beta cells. This genetic mutation alters the zinc transporter 8 protein and is associated with reduction of insulin secretion but not insulin resistance. These SNPs impact the risk of developing type 2 diabetes by creating a situation in which the body is not producing sufficient amounts of insulin and not regulating the amount of glucose entering cells. The genetic variations in certain genes can show how a person might be predisposed to having type 2 diabetes based on certain risk alleles in those specific genes. For example, in the SNPs RS7903146, the normal allele C can mutate to T, which is linked to diabetes. CT is 1.4 times more likely to get type 2 diabetes, and TT is 2 times more likely. RS1326664 and another SNP is in which the normal allele T is changed to C, increasing diabetes risk for both CT and CC. With so many variations linked to type 2 diabetes, not all of them are understood. For example, scientists do not know the function of SNP RS7756992, which is located on the gene CDKL1. Since 74% of risk of developing type 2 diabetes is environmental, a person who receives a test saying that they have very little genetic predisposition may still have a high chance of developing the disease if they make poor diet and lifestyle choices. A positive test might not always predict this trait because there are many genes related to type 2 diabetes, which only slightly increase the risk of diabetes. For example, a person with the heterozygous RS7903146 allele is only 1.4 times more likely to develop type 2 diabetes. A SNP test for diabetes would be about $200, which is relatively inexpensive. That said, there could be consequences of this knowledge. If a person is predisposed to type 2 diabetes and their insurance provider is aware of this, it can make their insurance more expensive. Having to pay higher rates for a disease one does not have and may never develop would be a huge issue. There is also an emotional burden from knowing you are at risk of developing this disease. One might constantly worry about developing diabetes in their daily life, which would be unnecessary for a person already making healthy daily decisions. However, this knowledge can also serve as motivation for someone living an unhealthy or sedentary lifestyle to make healthy choices that combat their chances of developing type 2 diabetes. Here are some references. Thanks for watching.